Hi, my name is Bill Zhang and I'm a junior at Homedale High School. My project aims to investigate the role of PAR3 in dendritic spine morphogenesis in CA1 hippocampal neurons in vivo. I wish to then translate my findings into the broader context of aging and neurological disorders. The United Nations estimates that the global population of people over the age of 60 is expected to surpass 2 billion by the year 2050. Now this number is nearly 3 times today's population. With age being a primary risk factor of all neurological disorders, and with an increasingly elderly population, it is important now to elucidate the biological proteins and pathways involved in neurodegeneration in a context of aging.
Dendritic spines are small actin-rich protrusions on neuronal dendrites that receive most excitatory inputs from synapses. Previous studies have demonstrated that a disturbance of physiologic dendritic spine homeostasis, such as spine loss, are common in neurological disorders. Moreover, cross-sections of spines reveal that a wide variety of proteins, ranging from neurotransmitter receptors to scaffold proteins, reside within a postsynaptic density of the dendritic spine. The presence of actin and other signaling molecules further demonstrate the organizational complexity of dendritic spines and their importance in the neural connectivity changes in development. My project's areas of interest will be the apical, which is emerging from the apex, secondary and tertiary dendrites and spines of pyramidal neurons within the SR layer of the CA1 region of the hippocampus, a temporal lobe brain structure that regulates several important processes such as motivation, learning, emotion, and memory. Reasons for deciding on studying this demographic are twofold. First, the SR layer of CA1 pyramidal neurons have been highly studied, as opposed to other parts of the hippocampus, and so it is most logical to collect data here for comparison with other conditional knockout animals. Second, the area between a soma, which is the cell body, and primary dendritic branches are not homogeneous in regard to dendritic spine morphogenesis, thus making collected data in those regions largely unreliable. Now moving on to the project's gene of interest, PAR3. PAR3 is part of the partitioning-defective polarity class of proteins, a group of highly conserved proteins that function throughout development to regulate cell polarity and asymmetrical cell division. Of these, PAR3 and PAR6 are specifically localized in the anterior cortex to combine with atypical protein kinase and cell division control protein to form what is known as a PAR complex. The PAR complex signals to numerous pathways, making them a major influencer in several essential cellular processes. Neurons are one of the most compartmentalized cells in the human body. Even more so are the spines of neuronal dendrites. The asymmetric nature of dendritic spines suggests that proteins regulating cell polarity may perhaps be involved in their formation. These considerations prompted me to examine the role of PAR3 in brain development by quantitatively assessing the length and spine density of the SR layer CA1 pyramidal neurons of the hippocampus of PAR3 conditional knockout mice. Next is my experimental design, outlined in the following table. Important to take note of here are the trials and the test groups. In total, I analyzed the spines of 24 secondary and tertiary dendritic segments. To avoid any conscious or unconscious bias, the number of wild-type and knockout brains were kept under blinding conditions. But the numbers you see here are post-data analysis. The two main test groups pertaining to dendritic spine morphogenesis are the wild-type mice, which acted as a control group, and the tissue-specific knockout mice, CaMKIIalpha-Cre. With that being said, the alternate hypothesis of my experiment is that the conditional deletion of PAR3 in postnatal CaMKIIalpha-Cre mice will promote dendritic spine morphogenesis and/or proliferation. Generation of tissue-specific PAR3 conditional knockout mice where critical PAR3 exons were flanked by LOX-P sites was carried out by mating homozygous FLOX-PAR3 animals with CaMKIIalpha-Cre mice. Essentially, the enzyme Cre recombinase is expressed under the control of a tissue-specific promoter, and 2 LOX-P sites are inserted in the introns flanking an exon of PAR3. In the targeted region, the hippocampus, Cre recombinase will be expressed and will delete the DNA sequence between LOX-P sites, thus inactivating the gene. In all other tissues, Cre will not be expressed and the gene will remain active. At around week 5, mice were deeply put under anesthesia with isoflurane prior to collecting the brain. Upon its collection, the brain is placed in distilled water, then immersed in 5 milliliters of impregnation solution for 3 weeks in the dark. The silver nitrate in solution randomly impregnates a small population of cells, allowing sparse labeling for data analysis. Afterwards, the brain was snap frozen in isopentane on dry ice and stored at -80 degrees Celsius in foil. A cryostat was then used to create 200 micrometer thick sections and put on gelatin-coated microscope slides and stored in the dark overnight. Within 3 days, sections were stained per manufacturer's protocol, and sections were then covered, dyed overnight, and then sealed with nail polish. Images could then be taken using a Leica bright field microscope at 0.25 micrometer steps in a z-axis to create a z-stack. Next, I utilized two image processing programs designed specifically for scientific multidimensional images, ImageJ and Reconstruction. First, I created a desired z-stack in ImageJ by opening all of the collected and saved images obtained from Golgi staining and converting the 2D images into a stack. I then imported the z-stack settings and enhanced the contrast setting to view the multidimensional image with higher clarity and precision. I repeated this process 24 times. In selecting a method of assessment of dendritic spine density, there are several important factors to take into consideration: time taken, accuracy required, and material study. In the past, researchers quantified spine density as the number of visible spines per estimated micrometer of dendrite. However, this method overlooks the fact that dendrites are rarely perfectly linear, and many spines are not included because they are obscured by the opacity of Golgi-impregnated dendritic shafts. A mathematical expression that I use to take into account such criteria and provides a method of estimating the actual spine density is the Feldman and Peters equation seen below. Moving on to measuring the actual spine dynamics. First, I found an appropriate apical dendritic segment. Next, I used the Draw Line tool to choose the segment's start and endpoints. Then, I used the Draw Z-Trace tool to measure the approximated dendritic length. Finally, in ImageJ, I used the multi-point function to count the number of spines along a segment. These tools collectively allow for more accurate and precise numerical data collection. As mentioned in a previous slide, to avoid any unconscious or conscious bias, the experiment was done under blinding conditions. But after data collection, the number of wild-type and conditional knockout brains were revealed. For analysis, I conducted my experiment at least 3 times with similar results. To test if my results were significant, I used a t-test, which revealed a p-value of less than 0.01. Thus, my alternate hypothesis was supported. To elucidate the role of PAR3, we utilized a mouse strain in which the PAR3 gene was conditionally knocked out. The Cre recombinase enzyme effectively catalyzes recombination between the 2 LOXP sequences flanking PAR3. My study's findings reveal that on average, spine density in conditional knockout mice was significantly higher than that of wild-type mice. The brain's ventricular system is a series of connecting hollow spaces called ventricles, which are essential for the movement of cerebrospinal fluid throughout the central nervous system. A special lining along the ventricle walls is composed of ependymal cells. Pathological conditions that are characterized by ependymal cell defects include hydrocephalus, a birth defect that causes enlarged ventricles. Hydrocephalus-associated mutations that may further define its underlying mechanisms have yet to be found in either human or model systems. Here we made an unexpected discovery: the deletion of PAR3 causes hydrocephalus in a postnatal mouse brain. We hypothesized that this phenotype is due to impaired ependymal cell integrity. Throughout the study, several obstacles were encountered. First, there are no established methods for automated 3D neuronal analysis. Much of the data collection relied heavily on manual computation, which was time-consuming and subjected to bias by experimental error and fatigue. Additionally, due to the fragile nature of dendrites, it was easy for dendritic shafts to break from the soma during staining, thus making it difficult to distinguish the spines and other intracellular material. This problem was encountered in the image on the right. Nevertheless, by obtaining numerous images of the same neuron and finding the cumulative average of spine densities, I ensured that the margin of error was small, thus avoiding major inaccuracies. The novel aspect of the PAR3 biology uncovered by our work expands the growing list of known functions of this versatile protein. The deletion of PAR3 in the CA1 hippocampal region resulted in an increase in spine density. From this, we can extrapolate other potential mechanisms. For example, the deletion of PAR3 might perhaps compensate for the lack of mature spines by forming more new immature spines. Researchers can now begin clinical trials to test the efficacy and safety of drugs that block PAR3 activity, and these findings support the rationale for such trials. Regardless, it is crucial to provide more data and explore more regions of the hippocampus. Furthermore, it was found that the deletion of PAR3 in the nesting Cre mice resulted in hydrocephalus throughout the brain. Ependymal cells are often associated with congenital hydrocephalus due to its cilial defects. Thus, our findings suggest a potential role for PAR3 in mediating ciliogenesis. Such insights into the mechanisms leading to hydrocephalus, if proven, carry many implications in today's society. Ependymal cell defects are often observed in aging brains and are associated with Alzheimer's disease. Thus, elucidating the role of PAR3 in ependymal cells will help us understand the mechanisms of brain aging as well as Alzheimer's, especially since recent studies show a loss of PAR3 in the brains of Alzheimer's patients.
Thank you for your time and consideration. I hope you enjoyed my presentation.